Hi everyone. In a previous release, I have introduced a new and challenging field, that of precision medicine, dealing with a disruptive and innovative approach in medical practice, a new way to look at patients and disease, where we strive to find the most effective treatment for each patient as per the patient genetic makeup and other predominant factors. What is at stake here is likely to be huge. This emerging field will create many opportunities for young and ambitious scientists to come up with new drugs and therapies tailored to serve patients now and in the future. No doubt, any investment in this direction is expected to be rewarding, and the first examples of precision medicine have generated big incomes to the concerned company. The critical value of precision medicine goes beyond the sole benefits of the pharmaceutical companies. It's a new way of thinking, said to be translational, where scientists from different areas of expertise come together to work and develop new concepts and create new medical solutions. As the challenges are manifold and the expectations are high, many training programs on precision medicine have been blossoming everywhere in the last decade. Many universities have designed and offered master programs to train new generation of scientists to take part of this amazing journey of excellence, crossing the border and advocating the need to collaborate with all research centers and academic institutions with one major and noble aim to improve the quality of life of all societies. Young scientists will certainly have many discoveries in store waiting for them. However, and despite the general interest in this field, it seems that many programs called precision, personalized, or translational are no more than a mere attempt to link basic science to clinics. That's simple. And some of them are no more than a developed version of the so-called pharmacogenomics. Precision medicine or translational medicine is an interdisciplinary approach involving stakeholders and professionals from various disciplines like genomics, molecular genetics, bioinformatics, system biology, biotechnology, structural biology, and other domains such as mathematics, physics, chemistry, computer science, law, business, and many others. Today's presentation is about pharmacogenomics, that many mistakes with precision medicine and consider as an equivalent term. So what is pharmacogenomics? First of all, I'm Professor Fahd Nasr from the Lebanese University in Beirut. And here you can see my private website that you are all invited to visit. In fact, the website features a number of courses for graduate and postgraduate students, and even for physicians and professionals. So if you are interested, you can go there, subscribe, and you will be notified of the latest posts and publication I showcase there. So pharmacogenomics, in a nutshell, what is this discipline? Is the study of how genes affect a person's response to drugs. It combines pharmacology on one hand, the science of drugs, and genomics, which is the study of the whole genome, all the genes, their functions, their regulations, their network interactions. So the aim of this discipline is to develop effective, safe medications that can be prescribed based on a person's genetic makeup. So it's about patient safety, as you can see. Severe drug reactions have been reported everywhere, with tens of thousands of hospitalization each year. Pharmacogenomics may prevent these accidents by identifying patients at risk. Clearly, it will improve healthcare cost and efficiency. So let's start with the challenges of pharmacogenomics. Remember, it's about finding out how variations at the genetic level correlate with different responses to the same treatment. So this field is complex, technologically complex, but is highly dynamic at the same time. It will help moving on forward with precision medicine and find the adequate therapeutic strategies, taking into account the genomic differences, environmental influences, and the lifestyle of the patient. Naturally, many barriers are there, but all of them can be weeded out to make the transition to precision medicine as smooth as possible. As key components to this evolving discipline are genes, gene number, genomic structure, genetic variation, SNPs, indel, haplotype, as well as gene expression profiling. Therapeutics is another keyword corresponding to the compound, vaccine, antibody, small interfering RNA, long non-coding RNA. As a matter of fact, the long non-coding RNAs are involved in many diseases, including cancer. I will dedicate one presentation to speak of the role of long non-coding RNA in exosome-mediated intercellular signaling. And equally, we have the PK/PD profiling. Just to say a word about this profiling, as I told you, it can be difficult to predict who will benefit from a medication, who will not respond at all, and who will experience side effects. So pharmacogenomics will use genetic information to explain inter-individual differences in drug metabolism, what we call pharmacokinetics, PK, and physiology or physiological drug response, we call pharmacodynamics. So that way we can identify responders versus non-responders to a drug, and we can predict the efficacy and or toxicity of a drug. So I hope the difference between PK pharmacokinetics and PD pharmacodynamics is clear. So just to remind you for the last time, PK is defined as the movement of drugs through the body, where PD or pharmacodynamics is about the body's biological response to drug. So PK/PD analysis is an alternative way to conventional dose effect analysis of the drug. If we have to sum up and speak of pharmacogenomics, so we have here great opportunities to increase the value of the drugs that we are developing using genetics, to obtain better understanding of the disease, to help predict disease diversity, severity, onset, progression, to identify genetic subtypes for the disease, to help in the discovery of new drugs, targets to distinguish among the population of those subgroups of patients responding differently to drug treatment, and equally to help in the interpretation of clinical results. So here we have a figure showing an integrative approach of comprehensive information flow from the patient, shown to the left, the patient's genome, all the way to find viable clinical applications. So this is a typical data evaluation pathway. You can see to the left the patient, and then we have the technologies, the adequate technologies to generate data, and then we have the analysis and interpretation of the data to build testable hypotheses. So it shows clearly the momentous role of bioinformatics, all those algorithms applied in genomics and other omics discipline to analyze genomic profiles. I refer to genetic variants, gene expression patterns, alternative splicing, fusion transcripts, epigenetic modification, transcription binding factors, and so on. So we integrate this information into appropriate networks, gene, metabolic, networks to facilitate the mapping of framework for the individual or individualized and personalized treatment strategy. So what we have here on this figure is the pathway for the integration of omics data in cancer research and clinical application. I will say no more about that. I will provide an example, a case study of pharmacogenomics. This case study is about 6-mercaptopurine. 6-mercaptopurine, referred to as 6-MP, is a purine analog that is effective both as an anticancer drug and as an immunosuppressive agent. So we use it to treat leukemia and autoimmune disease. Now, 6-MP inhibits DNA synthesis by inhibiting the production of purine-containing nucleotides, adenine and guanine. Now, 6-MP should be regarded here as a prodrug that must be first activated to form those compounds we call thioguanine nucleotide, TGN, of which the one shown in the middle here, the 6-thioguanine triphosphate, is the major active metabolite. And here you can see on the slide the pathway converting the 6-mercaptopurine all the way down to the 6-thioguanine or thioguanine nucleotides, having clearly clinical benefits. However, too much of those thioguanine nucleotides could suppress the immune system. So if you can see on the top, you have here an enzyme that we call thiopurine methyltransferase here that can divert the 6-MP from the normal pathway to generate another compound that is going to be cleared from the body. So let's focus on this TPMT. This enzyme, again, that we call thiopurine S-methyltransferase, is responsible in part for the inactivation of mercaptopurine, as you can see here. Why is that? Because TPMT catalyzes the methylation of 6-MP into the inactive metabolite 6-methylmercaptopurine. So this methylation will prevent the 6-MP from further conversion into the active cytotoxic thioguanine nucleotide metabolite, having clear clinical benefits. So certain genetic variations within the gene, the TPMT gene, can lead to decreased or absent enzyme activity. And individuals who are homozygous or heterozygous for these types of genetic variations, for these types of alleles, may have increased level of thioguanine nucleotides. And it means that they have increased risk of severe bone marrow suppression when they receive this anti-cancer drug. So we have here the case studies showing to the left individuals that are responding differently to this anti-leukemia drug 6-MP, as you can see to the left. So the population is split into 3 subpopulations. The major one on the top in blue, we have in the middle and down in red. So let me just say first, let's consider that genetic variation. We have a genetic variation, that allele coding for partial, low, or no activity of TPMT. So people that are homozygous for the normal functional allele, those on the top, as you can see, the vast majority, will metabolize the drug quickly. For this, doses need to be high enough to treat leukemia. Individuals that are heterozygous for the genetic variant for this allele in the middle will metabolize the drug slowly and require lower doses to avoid the side effect. And those homozygous for the mutant allele down in red are expected to metabolize poorly the drug. So the side effects in this case may prove fatal. So only a very low dose should be given to the low metabolizers, or what we call TMP, T deficient patient. So naturally, all it takes here is to proceed with a simple blood test, just to determine, in fact, the activity of TPMT. So individuals will be given the doses that are tailored with their genotype. So you understand here that diversity in terms of response to medication is due to variation in one gene coding for the TP-MT enzyme. So since we have one gene here at the basis of the differential, in fact, response to medication, we refer to this, in fact, discipline or to this case study as pharmacogenetics instead of pharmacogenomics. In fact, the two terms are interchangeable. Let me say first, in terms of definition, pharmacogenomics is about how variation in one single gene can influence the response to a single drug. Pharmacogenomics could go wider. The definition could be how all of the genes can influence responses to drug. Still, these two terms, pharmacogenetics and pharmacogenomics, are interchangeable. So in this example of 6-MP anticancer drug, we had at the basis one gene, and variation in this gene could determine the different responses of individuals to 6-MP. However, phenotypes could be much more complex than that, and many genes can be involved. So on this slide, you can see two possibilities for complex phenotypes. To the left, you have few genes and environmental factors, each of them contributing, in fact, a large risk. However, to the right, you might have a gene network, many genes involved in the response. So together with the environmental factors, each will contribute, in fact, a small risk. So we have come to the end of this short presentation about pharmacogenomics. If you like the presentation, you would like to show support, you have any question, please leave a comment. If you have a specific request, a private request, a proposal for a future collaboration, please write me directly using this email. Thank you for your attention and see you next time. Goodbye.